Today's topic, we're going to look at studying area study 1 for unit 4, which is talking all about the immune system within our bodies. And before we can start talking about the immune system in our bodies, we need to basically talk about the fundamental mechanisms that are related to the immune system, which is basically the detection part. How does your immune system detect foreign cells? And also, what are they? What are the different types of cells that or non-cells that they are actually trying to detect? So, what are these pathogens, and how are they even detected in the first place? So, we'll answer it. Be answering both these questions in today's class. So, the learning intentions of today are that you guys will learn that there are you'll be able to define the two types of antigens relating to nucleated cells. You'll also be able to define and identify pathogens and whether it's cellular or non-cellular, meaning that we will basically know about most of the antigens, and we will actually differentiate them based on whether they're cellular or non-cellular, meaning whether they are made up of cells or made up of non-living components, right? So an example of which is something like a virus. In most scientific debates, in most scientific communities, there's still a lot of debate on whether viruses should be considered alive, because without a host cell, without a cell that it's infecting, a virus doesn't do a lot of the things that are considered to be alive. For example, it does not reproduce by itself, right? Viruses are incapable of reproducing unless they have infected a host cell, and then it's tricking the host cell to make more copies of itself. So in that sense, should it be considered alive? Because it's only getting activated when it infects a cell. So is it even alive in that sense? So there's still a lot of hot debate about that, and there isn't a clear leaning towards one or the other. But there is arguments on both sides on why it should and shouldn't be considered alive. So another thing we'll be doing is also defining what an allergen is in relation to your allergies. So if anyone in this room has allergies, you get a quick understanding of why and how it is something you have to deal with, right? So the study design will ask you to look at the initiation of an immune response. What is triggering or what is initiating that immune response, and how that has everything to do with antigen presentation. So this one word, antigens, these are the triggers for your immune system. Okay, think of it. If your immune system is a journey of 1,000 steps, first step begins with detection, and that is with your antigen presentation. Okay, and your body basically needs to know, since cells come in all shapes and forms. Some are your cells, some are the cells from outside. Your immune system needs to be very good at identifying which cells are yours and which cells are not yours. It doesn't have to be someone else's, it's just not your cells. It needs to be able to identify that. As long as it's not your cells, it will fall under the category of non-self antigens. We'll go into that in a bit. So there's two types of antigens, self and non-self, depending on whether it is a part of your body, your cells, then it will be self antigens. If it's not a part of your body, then we will call them non-self antigens. And they can be present on cellular or non-cellular pathogens as well, and how that will also relate to allergens. These are all your glossary tasks. So let's get into the basics of the immune system. So, what is the immune system? It is the system in charge of your body's defense against any pathogens that try to infect and cause a disease for you, right? So there are heaps of potentially disease-causing pathogens that can infect your body, and that means that your body needs to be able to identify these cells without any issues as well. Sorry. Things. Yes, correct. All right. So, and body also needs to be able to identify these pathogens even if they're not cells. So they use something known as antigens. And one of the easiest ways antigens is either as a security badge that all cells have, or a identity tag that all cells have. And sorry, not just all cells. So all pathogens and all cells in your body would have them on the surface of this uh, on the surface, and these are used as identity tags. And how it works is that your immune system is basically very good at recognizing the antigens that are present on your own cells. These are called major histocompatibility complex markers, and these are the ones that are your own cells will present to on their own surface as a way to make sure the immune system recognizes them as being self antigens or cells, basically the cells that make up your own body. So by having this one antigen, the major by having this antigens, so there's two types of them, major histocompatibility complex. We'll talk about the two. But by having these antigens, your body is basically your cells are basically signaling to your immune system that this is your own cell, right? It's a marker, it's an identity tag that shows to your immune system that this cell is a it's your own body cell, right? Then the second part is when it's a non-self antigen. Basically, anything that invades your body, be that a virus or a bacteria, they will have non-self antigens. Basically, in this context, antigens can also be taken as either being different types of molecules such as lipids, proteins, sugars, or DNA and DNA or RNA, whatever the so a lot of the things that are either a product of the pathogen or something it releases or something that's released as a result of it basically getting killed. These are basically any leftover molecules from the pathogen itself. They can be considered antigens. And these antigens are going to be immediately triggering an immune response because they are recognized as being non-self, right? They don't have the major histocompatibility complex markers that that our own cells have. So your immune system recognizes that as being a different type of cell or a different pathogen that is foreign to your body. And as such, your body will immediately trigger an immune response to go fight that pathogen or whatever it is, right? So at this stage, your immune system is not very complex. It's basically just checking every cell, seeing what antigen it has. And if it has major histocompatibility complex, it will usually let it go. But if it doesn't have those antigens and it has any other antigens other than its own body's self antigens, it will trigger an immune response. So this is the keyword here. It doesn't matter if it is also major histocompatibility complex. If it's not the ones your body makes, it will still trigger an immune response. And we'll come back to that in a bit, talking about how that actually plays into something we do and how it affects that outcome. Okay, so let's talk about self-antigens. These are basically present on the surface of our cells, any nucleated cells in our body. Basically, what does MH1 class is nucleated. Sure. I'll go back to the slide, right? These antigens, these self-antigens, they remain on the surface of the cell so that any cell that's trying to recognize these antigens will immediately pick up on those self markers and will identify them as being a cell in your own body and will not trigger an immune response. The major or the most important antigen that our body makes is the major histocompatibility complex markers. And there's two types that are in relation to each other into the immune system. The first one is the MHC class 1, so major histocompatibility complex class 1. This is basically the most common one and the most important one. This is on the surface of all the nucleated cells. This is basically all the cells with a nucleus. It will have these histocompatibility complex markers on their surface, and as a result, basically they will be recognized by any immune system response as being our own self. So that is how our body recognizes it most straightforwardly using the MHC class 1 markers, right? The MHC class 2 markers, on the other hand, they're a bit more different. They are basically used specifically on specialized cells of the immune system, and then they basically use this uh, marker to present any pathogens, antigens to the immune system. Think of it like someone bringing up evidence of a crime to the police, right? These specialized immune cells bring this antigen of the pathogen to specific cells in your immune system and presents itself to them, right? So these are cells that would actually end up presenting these antigens to T lymphocytes or stuff like that. And that's why they need this special antigen class with them, right? It's used to present the pathogenic antigen to the specialized immune cells. Because why is this needed is that in a general pathogenic attack, let's say it's localized to one part of your hand, right? So your immune system triggers a response to that part of the hand. But what happens is a part of your immune system will pick up antigens from that localized area and bring it to all over your body, looking for the right. I don't want to go into too detail until I teach about it, but basically, there's an immune response triggered called specific immunity, where your body is specifically making antibodies against that disease or that pathogen. And that can only be triggered if your body brings that antigen and goes and looks for that uh, B lymphocyte that is able to make those antibodies and presents the antigen to it so that it can calibrate its production of antibodies towards the disease. And once that B lymphocyte is calibrated, it will produce massive amounts of antibodies that are specifically targeted to deactivate that pathogen. We'll get to that in the later topics. But for now, all you need to know is that MHC2 antigen is used to is specifically found on specialized cells of the immune system, and they are the cells that will present the antigen to other specialized immune cells for later benefit. Okay, so now let's talk about the non-self-antigens. These are basically antigens that trigger the immune response, and as such, they will basically be any antigens that are not found in our body. So our body makes a very specific type of antigen, major histocompatibility complex, and it also makes it specifically to us, right? So in between each other human, our major histocompatibility complex will be recognized as being foreign if we are not doing our prior research, right? So we're going to get into why that's an issue. So this system of using non-self and self antigens relies very heavily on the body's ability to correctly identify self antigens from non-self antigens, right? Only if your body is very good at telling which cells in your body are yours and not yours can this system of using antigens work efficiently. And most of the time, that is the case. Most of the time, your body is really good at identifying self antigens versus non self antigens. But there are also places where this becomes a issue, and we're going to talk about that. So just to give an example of how this non-self antigens would end up triggering an immune response. So example is when a bacteria is infecting your body, right? You've had a cut, bacteria is going into the cut. All these bacteria are trying to consume nutrients and reproduce and all that, and they have specific surface antigens that are foreign to your body, that are non-self antigens. As soon as a cell detects the fact that it doesn't have the right antigens, it will immediately send signals to your immune system to trigger an immune response. As a result, your body will basically be aware of the pathogen in your body and will start to trigger systems to make sure to combat it. So this system of ensuring any foreign cells in our body is always found out isn't always a good thing, right? Sometimes we actually want that foreign cells in our body. A few examples is when we have organ and blood donation. So one thing to note here is, right, even with blood donation, we still have to worry about antigens, even though they're not nucleated cells. Remember that I mentioned that these MHC class 1 are only expressed in nucleated cells, and are red blood cells nucleated? The answer is no. Red blood cells do not have nucleuses, so they do not have MHC class 1 markers, but they still have antigens present on them. That's why they are also regarded as being dangerous if you're from different blood groups, right? If you're not from compatible blood groups. So let's talk about organs first. So with our organs, there's definitely an MHC class 1 marker antigen on them. And that means that they are also unique between different individuals, right? We all might have MHC class 1 markers between us, but they're still different between different populations. And sometimes it can be so different that you literally have to look for a perfect match before you can have it, because if you don't have a perfect match in terms of your antigens, then what can happen is that the antigens from the donator will be different compared to the antigens inside the body of the recipient. That means that the MHC1 class markers, that's right, class 1 markers on an organ from a donator will be triggering the immune response from the recipient's body, because it recognizes it as being foreign, right? So an example, let me paint a picture. So the person who's donating, person A, he is having different antigens compared to person B. He donates his liver. As soon as the liver is transplanted into the donor's recipient's body, the markers on the liver are recognized as being non-self by the recipient's body. Basically, the body's normal immune system is moving around, and when it gets the liver, it picks up on the fact that the markers on the liver cells are different than not the usual self antigens. There are non-self, sorry, non-self antigens, and as a response, the immune system will get triggered and it will start to attack those cells, basically attack a whole organ. And this is why, if you do not carefully make sure that the antigens are going to be compatible, your body can literally destroy that organ that you were just given to save your life as well. So this is also why people who've recently gotten a organ transplant do take immunosuppressants, because they need to make sure that their immune system is not fighting the new organ. So sometimes they do have to take immunosuppressants to ensure that that doesn't occur. Any questions about this? All right. So now that we talked about the MHC1 class markers on organs, let's talk about the antigens on blood and how that relates to blood donation as well. So we need to make sure that we are aware that the antigens on blood have nothing to do with the MHC1 class markers. Okay, there are no MHC1 class marker antigens on blood. There's only two types of antigens on blood. One is the AB antigens and the RH factor antigen. So the AB determines what kind of letter it has, A, B, or O. And then the RH factor determines whether it has a positive or a negative, right? So if your blood type is O positive, that means you don't have A or B antigens and you have the rhesus factor present. So that's your antigen combination. If your blood type is B positive, that means you have the B antigens and also the RH factor. You have the RH factor, that's why you're a B positive, right? So let me zoom into the picture, maybe that will make it better. So there are essentially three, technically there's only two groups of antigens. The RH factor is one group and the A and B antigens are one group, because A and B will determine what letter it has, whether it has both antigens, one of the antigens, or none of the antigens for the letters A and B, right? The RH factor is another type of antigen, and that determines whether it has, basically whether it has that RH factor or not. Positive means that it has the RH factor, negative means that it doesn't have the RH factor. Okay, does everyone make sense of this chart? You don't have to remember all of them, as in like you don't have to tell me what if A plus, what does that mean in terms of the antigens it has. It's just that you need to be aware that there are multiple antigens relating to blood type and how they function, right? So talking about this, let's put example into the different blood groups and how that works between each other, right? So why is it that A positive blood type cannot just donate to a B positive blood type? What do you think? Right? It's basically has to do with the antigens present on the cell, on the red blood cells. So let's say you have A positive, right? That's your blood group. That means that you have the antigens for A present on your blood group, and you also have the RH factors present on your blood group, right? Those are the antigens that you have present. So if you donate this blood to a individual with B plus so if you donate, if some don't need to overthink it with the rhesus factor. So now let's just use the letter groups. So let's say A donates to B. What are the issues, right? For example, what would happen is that it has A antigens present on the surface of the blood group. Then it's being donated to the B. So what happens is that the the immune system in B, the person who has a blood group of B, will not be able to recognize these antigens, right? As soon as that's occurring, it will say these are non-self antigens. They're basically foreign antigens that are present on the blood group. That means it is a pathogen that's trying to infect our body. So this blood group will be recognized as being a threat and will activate the anti-A antigens, antibodies in your plasma, right? Basically, if it's recognized as foreign, it will basically immediately assume it's the pathogen and will trigger an immune response. Same thing with B going into A, right? Blood group B, if they are donating to A, blood group A, blood group B has antigen specific to their blood group, and if it is transferred over to a person in A, it will be recognized as being non-self antigens, and it will trigger an immune response with antibodies that are basically gonna target the B blood group. So you can imagine how dangerous that is if it's your blood, because your blood is going to be moving around all over your body. And if your immune response is getting triggered to all parts of your body, it is much worse on you. All right. So now let's talk about the interesting case with AB, right? AB blood group is a specific blood group that has both types of antigens present on its surface, right? So what does that mean is that it could basically receive a blood from any of these blood groups, right? AB, or O. Let's talk about why. So the antigens on this cell, on this red blood cell will be still recognized by the cells that are detecting antigens in the. Okay, let me try to draw this out, not be less confused. Okay, so let's say the donor is someone in A and the recipient is someone in AB, right? So we have to consider what type of antigens does this donor have, right? They have antigen A. So if it has antigen A, for the recipient to not get triggered, not trigger an immune response, they also need to have that same antigen. And as we can see here, AB has the antigens for A in its own red blood cells. So if it detects antigen A when it's donated, it will be able to actually determine that they are self-antigens because of the fact that they have the same blood group A antigens present on its own blood cells, red blood cells as well. All right. So that we've talked about A. Let's try mentioning a different example. So let's try B, right? So if you try B, it's the same thing, right? What type of antigens it has present on the red blood cells will be antigen B. And why will that not trigger an immune response in the recipient? Is because they also have the B antigens present on their cell. So their body will see the B antigen present in this from this donor and say that that is also something from our body. It will not interpret it as being pathogenic or foreign because it has the antigens that are your body uses to recognize it as being self or non-self. Yes. So AB cannot donate to A or B because of the fact that they have that counter antigen present on them as well. So that will trigger the immune response of one or the other because they basically have the counter of each other, right? So if AB is donated to person with B, then the antigens, the A antigen on the red blood cell will trigger an immune response to this person. If AB is donated to A, then the B antigens on the red blood cell will trigger an immune response in that person, right? So it's convenient that they have the antigens and the body or the recipient will recognize those antigens as being self, so it won't trigger an immune response, right? So, okay, now let's go towards O, right? Let's try donor O, any blood type. So if AB, specifically AB positive is a universal recipient, that talks about the rhesus factors. But essentially, for your level, you guys have to really focus on the rhesus factors a lot. You don't really mention it even in your textbooks. So I'm not going to put too much energy into it. Just know with the blood type A, B, or O, there are specific that are present or not present on the red blood cell that will trigger specific immune responses from one another, right? So lastly, let's look at O and how it relates to the other blood types, right? If AB is the universal recipient, O is the universal donor. Basically, they can donate to any blood group they want because more importantly than anything, they don't have any antigens present on the red blood cell, right? So essentially, this will go undetected by the cells looking for antigens because it doesn't have any antigens present, right? Essentially, antigens are the detection system, right? The body looks at the antigen and determines whether it's a pathogen or not a pathogen. So if you don't have any antigens present specific to blood groups, then it won't identify it as being foreign or self. it will just not detect it, right? It will go, consider this almost like an incognito mode, right? When it has no antigens, it's incognito to your own immune system because it's not triggering an immune response, neither is it thought as being your own cells. Anyone confused about this idea? So that's why O is capable of receiving blood groups. That's right. Is able to be a universal donor because it has no antigens in the red blood cell present on its surface. Any questions? So let's think of an example, right? O recipient is AB, right? So this one does not have an antigen because it's a red blood group of O, and O does not have any antigens present on the surface, right? This person is donating to AB, right? So when the blood from donor O goes into the person in AB, it's their cells will start examining these red blood cells. And that's when the cells realize that these red blood cells don't have any antigens present on the surface, and as a result, it won't trigger an immune response because it will only trigger an immune response if it detects the non-self antigen, right? Basically, an antigen that is not associated with your body. If it doesn't have any antigens, it's basically not going to be detected at all. It's just going to let it through, right? So you can either, so there's two ways you can survive when donating to a certain person. One is to make sure that you have the same antigens as them. Or second is to have no antigens present on the cells at all. And this is only something you can do with blood groups because of the situation where there's a blood group of O with no antigens present. With your cells, you can't really do that because there's the MHC class 1 markers present on all nucleated cells, and you can't avoid that. So, all right, let's see if I can get you guys to have a guess at that. Let's play a small game where I give you two blood groups and I ask you guys whether you think they can donate to each other. All right. So donor is blood group B. Okay. Recipient is blood group O. Do you think this is gonna be okay, or do you think it's gonna be bad? All right. Who thinks it's gonna be okay? Raise your hands. Who thinks it's gonna be bad? Raise your hands. Yeah, it's gonna be bad. Okay. Let's point it out why. So first thing to remember, right? You'll do best to remember these two phrases, right? So these two statements are very important to remember. Blood group O is a universal donor. That means they can donate to anyone they want, but they can only receive from one blood group, which is O themselves, right? blood group AB is a universal recipient. They can receive from anyone, but they themselves can only ever donate to AB, right? It's basically vice versa of inverseness between each other, right? Because this one is a universal donor, it can only receive from O. This one is a universal recipient, that means it can only donate to AB, right? Make sure you guys are aware of that. So let's look at it in the context of why is that the case, right? So we have blood group B who is donating to blood group O. Why would this not work out? First thing we ask ourselves, what antigens are present on their respective cells, right? This one will have antigen B present, and this one will have no antigens present. So suddenly there is a new antigen that's going to be present in the recipient body, and the recipient body will now identify it as being non-self-antigens, and as a result, it will trigger an immune response, right? When you're thinking about how it will affect the donor or the recipient, you must ask yourself, what are their antigens right now? What antigens do they have? Do they have B, A, or both A, B, or no antigens at all? And whether that will trigger an immune response in the recipient's body, right? B has the antigens, and as such, the recipient will then identify it as being a non-self antigen because its own body doesn't have any antigens for red blood cells. So suddenly, if a red blood cell had an antigen that it didn't even recognize, it will say that these are basically pathogens trying to infect our body, and an immune response will be triggered. I know this can be a bit confusing, but if you play the quiz of trying to understand who can donate to who, this kind of makes more sense over time. So maybe just try to create a chart and try to label it yourself manually of who can donate to who, and then have a reasoning in your own head of why. The use of antigens can also sometimes be faulty, right? Not just it's not a perfect system that is always going to work. Sometimes it itself can be faulty, and that can come in the expression of something known as a autoimmune disease. So you guys might have heard of the phrase autoimmune, or it might have been been mentioned to one or two of your friends. But it's basically uh, the fact that your own cells that have self antigens are recognized as being non-self antigens. Basically, your body has started to misidentify your own cells as being foreign cells, and will then trigger an immune response to target and attack your own cells, right? This is basically a situation where, for some reason, the immune cells in your body will start to recognize the antigens present on your own cells as being foreign, and will start to attack them and destroy them. And these can cause a lot of different types of diseases that you guys have probably seen or heard of, like eczema. If you've noticed eczema, it looks like your body is peeling off its own skin, right? That's because it really is, right? Your body is basically identifying the skin cells as being foreign, and will basically try to trigger an immune response to get rid of them. That's why there's a lot of redness and inflammation in the eczema areas, because an immune response is being triggered, and that is a part of a autoimmune disease. So another way it can also be faulty is with your allergic reactions. So allergic reactions are caused by something known as an allergen, right? People who have allergies would probably have heard of the phrase. But an allergen is basically a non-pathogenic antigen, meaning that it is not a pathogenic, meaning it's not a disease causing antigen that is triggering an allergic reaction. So that is basically something that is harmless, but your body thinks as being very harmful. Specifically, how do I describe this? Is that any of these different things, right? Very unrelated things. People are allergic to a lot of different things out there. So anything you're allergic to, your body basically detects these antigens present on those whatever they are, those items, and then it will basically decide that this is a very dangerous and very harmful substance because of it having these antigens, and will trigger an allergic reaction, which is a inappropriate immune response to the allergen. So think of something specific like eggs, right? Food allergies are very common. Then food allergies. So let's say you're allergic to egg. What happens is that when you are exposed to these eggs on your skin or inside your body, the antigens, this food will be triggering an immune response in your body. And that response is called an allergic reaction. Why is it called allergic reaction? Because that's talking about an inappropriate immune response. Basically, it's too great of a response for something so small and non-pathogenic. Basically the equivalent of deploying the army to quell a, I don't know, gas holder, a gas station holder, right? It's super inappropriate and it's super overkill. It's the same thing with your allergic reactions, right? Even though there's a small exposure of a small peanut, right? You touch it with your skin, sometimes people can get very, very bad symptoms like rashes, runny nose, their throat starts to close up, their shortness of breath, and also sneezing, and so many other things. And it's all caused by your body basically detecting certain traces of antigen in these items, and then that basically being deciphered by your body as being incredibly dangerous, and that triggers a allergic reaction, which is something that can really take over your body if you're not careful. Yes. Friend has like really vitamins. And they also have asthma. Does that mean like I might develop that other stuff? No, no. Like so autoimmune diseases, they're not exactly linked to each other in the sense that you get one, you get others, or is that it's a progression thing, I think. It's a very specific case of cells in that body being recognized as being a non-self due to the situations that are specific to that region. So this is something that's not supposed to be happening, right? Why could it be happening? Stuff like mutations within those specific cells that are making them cause a autoimmune disease. It can also be caused due to genetic factors, but it doesn't mean that they're all related to each other. There are very specific cases that triggers eczema. There are very specific cases that trigger asthma. So it has to do with the antigens and also the corresponding response by the immune system. How extensive is the response, right? If it's something as bad as rheumatoid, I remember correctly, rheumatoid is when your, what's that, arthritis? Uh, I'm getting. So one of these autoimmune diseases is when it is triggered to basically attack the cartilage in between your bones, and that means that you're basically going to be having trouble moving the longer you have it, because eventually it will start to make your bones grind against each other. So the prominence of these autoimmune diseases within an individual is always changing. It's not fixed, right? Sometimes it can actually get better, sometimes it can actually get worse. And that all depends on the immune system and the corresponding antigens present on that specific cells. If they're going to be recognized as being non-self forever, then they will have those autoimmune diseases forever. But if eventually somehow your body starts to actually correctly recognizes the antigens on these on our own cells as being self instead of non-self, then the autoimmune disease won't necessarily be prominent anymore. I'm not familiar with all the types of asthma, but there is asthma caused by an autoimmune disease as well. Right. So now that we talked about how it's detected in the body, right? How it's detected by the immune system, how are pathogens detected by our immune system? Let's look at our usual suspects, right? So we have two categories, broad categories of pathogens, basically disease-causing entities, and they can be either cellular or non-cellular. Basically, they can be considered alive, living or non-living acellular, right? Their damage can either be extracellular or intracellular as well, meaning that they can do their damage outside of the cell in the external environment of the cell, or they can basically go into the cell and also do damage, right? It all depends on the type of pathogen we are dealing with. So let me give you an example. So first one, we have our bacteria. So these are unicellular prokaryotes that can basically infect any part of your body, right? They are basically all over you right now, right? Bacteria is covering you, it's even inside your body right now, and they can sometimes even be beneficial. But if exposed at the wrong part of the body, or if they are basically pathogenic, they can cause a disease through the production of toxins and enzymes, which basically counter the functioning of a cell or can even cause it to die. So one thing to note is that bacterial cells, they will typically reproduce asexually via binary fission, right? Basically, this is the process where one bacterial cell will accrue enough resources in itself, enough food that it can split into two identical, genetically identical cells that are just copies of each other, right? So there was one cell, it grew enough that it can split into two cells, and both those cells are genetically identical with each other. That's why it is considered as an asexual reproduction, right? It doesn't need another partner. It just does it on its own in the sense that it makes a genetically identical copy of itself. And these can cause all kinds of diseases. For example, that's your, I can't pronounce this, right? Neisseria, meningitis, bacteria causing meningitis, or the Clostridium tetani causing tetanus, right? There's a lot of bacteria out there. Each of them can be so a lot of them can be pathogenic depending on where they emerge in your body. All right, next is your fungi, right? These are eukaryotic microorganisms that are potentially pathogenic. So what's the difference between eukaryotic and prokaryotic? Is eukaryotic is a complex cell with organelles like mitochondria, chloroplast, and many more. Whereas prokaryotes are a simple cell that don't have these special things like mitochondria and stuff. So that's something that will always be important to know in overall biology that will be mentioned a lot. So make sure you guys remember that. So fungi are eukaryotic organisms that include stuff like yeast and molds, and they basically look like a branching filament called the hyphae. So it looks like a tree branching out, and it's one of the only microorganisms to do it like this, where the fungi branches looks like it's branching out like that. And they basically have two forms of reproduction, asexual or sexual. For sexual, they will reproduce through spore formation, and with asexual, oh sorry, they will both reproduce both through asexual reproduction and sexual reproduction via the spore formation. A few examples is athlete's foot, that's caused by fungi, ringworm. That's basically something that can potentially happen if you don't wash your socks for a long time. You keep wearing the same socks. You can have something in a ring shape forming on your feet that is caused due to a ringworm, if I remember correctly. Then there's also thrush. We can basically go through the different diseases caused by these different pathogens in a bit. So now let's look at another cellular pathogen, which are your worms, right? These are scary buggers, and these are not microorganisms anymore. They can actually grow pretty big. In fact, the longest one ever found inside a human body was 55 meters. Yes, it's meters. These things, an example of one that you guys probably would have heard of is tapeworms. Who's heard of tapeworms? So tapeworms, one of the most common places they're found is in your intestines. And your intestine basically coils around a lot, right? Imagine a tapeworm that's just growing inside it. Yes.
Did you get those, what you do with kids to remove worms in the farm?
Yeah, worm medication. Why would I say? Isn't that? I can't remember whether I did it, but even if I did, I probably wouldn't mention it because it's yeah. Okay, just to scare you guys a bit, I'm gonna show you a video of a tapeworm. Okay, okay, not it being extracted, but just tapeworms. No?
It was.
No fun.
I don't think the exam will show us.
It won't. No. With the tapeworm, think of it like any not say insect, but basically something very similar to an insect. They basically reproduce sexually via a complex life cycle involving an egg followed by a larva or larval, and then your adult stages. So this is very important to consider. They don't just emerge in the small intestine. In fact, they can emerge anywhere in your body, depending on where the larva rests, where it hatches, and adult larva grows, right? Adult tapeworm grows. So, some of the most unorthodox places that tapeworm have been found in the brain. It's not supposed to go up there, but it can go up there if the situations are correct. In muscles, in the small intestine, that's normal. But other than that, yeah, it can appear in a lot of different places, and it's pretty interesting to think. And yeah, how can you get them? Basically, drinking unfiltered water. So be careful when you drink unfiltered water from rivers and stuff like that. Not just tap water. Tap water is fine because it's being treated at a plant. So they make sure they don't have any ringworms, anything like tapeworms and stuff. But if you go to outdoors, rivers, lakes, be very careful about what you're drinking. Make sure you boil your water like extensively before drinking it. Yeah. I mean, I don't know what am I looking at. Like I assume that is a sample. But yeah, basically uncooked, unfiltered, anything raw, be careful of that. You're not even showing any fun things. Let me play a video. See it. Yeah, Google it. I can't Google show it to you without showing everyone. Okay, sure. So yeah, example, here is a brain tumor that is essentially a tapeworm. So doctors can even misidentify them as being brain tumors and then later realize, no, no, that's an entire tapeworm there. So complex medical procedures. They cut open your skull, operate, take it out, like pulling a string out of your body. In fact, if you look at the procedures with like your intestines, that's how they do it. They're like pulling noodles from like a bowl or something. They just flush. It's pretty funny. Okay, moving on. Nice. Okay, next cellular pathogen is your protozoa. These are single cell eukaryotes that are can be free living or as a parasite, and they have different mechanisms for action. So some of them are acting as cellular pathogens where they actually cause problems, inhibit nucleic acid synthesis, protein synthesis, and various stages of cellular respiration, or they essentially just become a parasite and just extract resources from their host. So they can reproduce through asexual reproduction and sexual reproduction. An example of that is Plasmodium, which causes malaria. So, important thing to note, right? So with malaria, this protozoa is causing it, but people associate what's the word? Mosquitoes with malaria, right? So mosquitoes in that sense are the vector. They are the one that brings the pathogen to the living thing. But the pathogen is basically what is causing the disease. So never get confused about what the vector is and what the pathogen is, because vectors are what bring it to the organism. A pathogen is basically what is causing that disease. So always know that. And then viruses, this is our first of non-cellular pathogens, basically the ones that are not considered to be living, right? When we deal with viruses, they are basically one of the uh, most weirdest and curious things about basically life, I would say, because they are the boundary of what is considered to be alive and not alive, because we can't exactly determine whether they are alive or not, because they are very good at carrying out instructions in their DNA code or their RNA code. But other than that, we don't really see any uh, interactions going on with the virus, right? So this is basically the case where we have a infectious agent which is basically just made up of DNA or RNA. Not all viruses have DNA even. Some of them are so simple that they only have RNA inside them, and that is capsulated inside a capsid, right? Protein code called a capsid. And when that uh, when that virus cell is entering into a host cell, it will release this genetic material, and that is going to be interpreted by the host cell as being genetic material that it needs to use. So when it uses it, it makes more of the virus, and that eventually causes a loop that can eventually lead to the host cell being killed, right? Viruses can cause disease through the lysis of cells during viral replication. Basically, that's the final situation that happens when this process goes on over and over, right? The virus tells the cell to make more copies of itself. It keeps doing that over and over until all the resources inside the host cell have been taken up, and there's literally no more space for the virus cells to go, virus particles to go, so they literally burst the cell open. That's what we call as being lysis of cells. Lysis is basically, think of a water balloon that pops. That is what lysis should look like to you guys. So essentially, when lysis of a cell happens, all of the insides will flow outside. So viruses love that, because when that happens, they can get exposed to a bunch of more host cells, and they do the process all over again. So they also are responsible for formation of some cancers by affecting the gene expression, and also can lead to the overstimulation of the immune response that can then lead to organ damage. So the most important thing of why viruses are not considered to be alive is because they are not able to independently reproduce, right? They have to insert their genetic material uh, into their host cells, and then use the protein machinery within those cells to make more virus cells. Right? Examples of these viruses: rhinovirus, influenza, Ebola, SARS-CoV-2, COVID-19, you know that. COVID-19 is the disease, SARS-CoV-2 is the pathogen that causes the disease, right? There's a important distinction to be made, because if someone said that COVID-19 is the pathogen, you'd be marked wrong. It's the same thing as saying malaria is the pathogen, you'll be marked wrong. These are examples of the disease that the pathogen is causing. The pathogen itself is talking about the cell or whatever that is causing that disease, right? So viruses, influenza would be the virus, flu is the uh, disease, right? Ebola is the pathogen and it causes, right? Ebola virus is the pathogen and it causes the Ebola disease. That's one way you can get away with both. So just use Ebola. That's most straightforward. All right. Now, last non-cellular pathogen is your prions, right? These are basically abnormally folded proteins that have the ability to induce normal proteins nearby to become misfolded, right? They basically induce normal proteins that are folded to specific functions to become misshapen, and that means that they are not able to carry out their original function, right? So they only occur in mammals and affect only the brain and other neural structures. They are currently the only known infectious agents that don't contain nucleic acid. Basically, only pathogen that is known to not have any genetic information, right? Even viruses, which are argued to whether be considered alive or not, have genetic information, and that's why they can program cells to do whatever they want, right? They basically give the cell instructions and the cell just does what it's told. But with these prions, they basically are molecules that are abnormally folded, and they can then induce other normally folded proteins to become abnormally folded or misfolded, and that can have a detrimental impact. So they do not reproduce in the normal sense of the word. Instead, they induce folding of nearby proteins, therefore spreading throughout tissue. Think of it like this. Uh, yeah, most straightforward way. Think of it like a forest fire that starts from one tree, right? That one tree, that one protein is misfolded, and every time it interacts with another protein, it will misfold that protein, and then that becomes a prion itself, right? A forest fire starts with one tree, that fire spreads to another tree, and then now there's two trees that are on fire that can spread that fire further, right? Taking the prion, it starts with one misfolded protein, which then interacts with another protein, and it's going to be an exponential growth from there. It's the same thing as one bacteria reproducing to become two bacteria reproducing to become four reproducing to become eight to 16, 16 to 32, and so on, right? Same with the prions. They can have an exponential rate of growth depending on the number of proteins they are interacting with. So far what we have learned today, right? Essentially, there's two parts to what we have learned today. If the topic today was detecting pathogens, the first thing we looked at is how are they detected in your body? Okay, what is doing the detection system? And that's where we have our antigens, right? Self-antigens are basically your body's antigens that your body will recognize as being your own. Okay, these are basically badges that your body gave to every cell in your body to make sure that they know that it's part of your body, right? And then there is non-self antigens. These are basically badges given by other immune systems that will immediately get recognized in your body as being non-self, right? They are basically a telltale sign of being a foreign cell. And based on whether it detects non-self antigens, it will, sorry, if it detects non-self antigens, then your immune response immediately gets kicked into action, and they are going to be starting to look for those non-self antigens and where they are, where the source is, right? So since we've talked about how it's detected, let's talk quickly about the pathogens themselves. There's 6 different kinds that you guys need to be aware of. There's 4 which are cellular in nature, meaning that they're considered as being alive, and then 2 which are considered as being non-cellular, which are not alive. Out of the 4, protozoa, fungi, and bacteria are microorganisms, and worms usually grow to be as tall as 55 meters plus. Sorry, it can grow up to 55 meters as well. So worms can be pretty big, whereas protozoa, fungi, and bacteria usually are small bacteria, small microorganism infections. With your viruses and prions, they are considered as non-cellular because they're not alive. With your viruses, they carry genetic information within their enclosed capsid, and once they infect the cell, they release it and force that cell to to produce more of those virus cells. With your prions, that's misfolded proteins that then cause other proteins to become misfolded, and that starts a chain of misfolded proteins